telling you what it is. We are doing a problem analysis. We have a situation, and now under ZP and natural farming, we are looking at what is going on, how do we understand it, and how do we apply this. So this is really the sort of problem analysis that you as teams would be doing. And as Dee so correctly and importantly said at the beginning, it's not a hypothesis, but there's a lot that we don't know. You know, everything always, yes, we can be certain of these things, but there are also other parts that we have to sort of explore and find the answers to. So we can say, yes, what we'll put up 80%, yes, 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 but there'll be 20% where we have question marks and we need to explore. So it's good that you're asking lots of questions, and it's actually quite natural that there's a lot of red herrings amongst the questions. But that's okay. But so we've got to focus. But now we have a clean whiteboard, so we can go. This is marvelous. Right, so what did we do with pre-season, pre-monsoon sowing? You know, we want it because we want a green bridge, and what we had is a red soil surface, and what we did is we took some seed, mixed seed, and we covered it with biostimulant. So we broadcast seed with a biostimulant coating around that seed, didn't we? And it's from that we're trying to achieve 365 days green, which is a big challenge, not easy. Okay? And in a sense, this is how we understand, or I understand, but let's have the debate and argument, what could be happening. And it goes in a sequence of stages, and the sequence is very, very important, and it'll answer a lot of your questions. So the first thing is you have a biostimulant, and it will stimulate a whole lot of fungi in the soil. But these aren't mycorrhizal fungi necessarily, because these are just saprophytes. Okay, these are fungi that basically break down organic matter in the soil. You know, they just eat up some of that organic matter in the soil. And we've already more or less agreed that when we do that, we generate water. Okay? Because when we break that down, this will generate 1 gram, 6 grams of water in the soil. Okay? So we have no problems with that, and that's what Dee told us. Here is the biochemical water that's generated. And because there's no water from rain, you know, this is still sort of 4 weeks, 6 weeks before rain, we have to assume, again, this is 80% confidence, 20% question mark. So we're always open for good ideas. We have to assume that there's enough water here that basically cause that seed to germinate, which is logical. So now we have actually the first big lesson. If we have a biostimulant, it can break down, it can accelerate these saprophytes to generate enough biological water to get that seed to put down its first root. And obviously there has to be water for that seed to germinate, so it had to have water from somewhere. It hasn't rained, so this is a very logical, reasonable assumption. But of course this is now very, very, very risky for the plant, because it's put a radical down, it's put a little root down, and unless it gets water and nutrients fairly quickly, it's dead. Okay? So it's a gamble, because we could lose it all. So how do we keep going? Because we have a biostimulant, it's fair comment, the minute we have a root, we're going to have nutrients from the seed exuding out, and it'll be sort of creating its network of mycorrhizal fungi. And we know that. It's within days that these mycorrhizae colonize the root. And so these plants very rapidly form, and they can't actually colonize and survive without the mycorrhizal fungi. Okay? And this is then where the second stage of what Jehne was outlining steps in. Because you remember earlier we had that little diagram. Here is the soil, the healthy soil. And yes, it's got these voids. And this is where the soil water was. But we know that that soil water is gone because it hasn't rained. So there's no freely available soil water, right? Okay, that soil is at wilting point. It hasn't got, that's gone. So there's negative here. But we do know that there are water films around each of the soil particles, and these might be at 3 times wilting point, unavailable to plants. But as Jehne explained, that water is available to the mycorrhizal fungi, because the fungi can grow over these particles, and as Jehne explained, it has a membrane tube, and it can absorb that water across that membrane at 5-10 times wilting point tensions.
Can you explain wilting point tension?
Okay, a wilting point, water is in a soil, and there is this free water, or water that's available as we sort of in that black void, right? And this can easily drain, and in a sense plants can get access at that water. But as Jehne showed in a bottle with a straw, there's other water that's sitting around these soils on films, and they're held much, much more tightly that plants can't get. They're beyond the wilting point or the suction potential of that plant. But the fungi can get those, so these membranes, these fungal membranes, microscopically growing over those surfaces can. So in a sense, these plants can give the plant water. And we haven't got another colour, doesn't matter, we can do it in black. These fungi can also, of course, take up nutrients from the calcium and the magnesium and the phosphorus that's on those surfaces, right? By the same membranes, these are actively absorbing the nutrients into the things. So not only have they got water, they have nutrients. Okay, and this is really critical because the survival of this plant or this seed depends on, can it, in its first 5 days, get roots down to depth? Can it establish connections to water and to nutrients from the soil before its reserves in the seed run out? Very critical, right? It's a very competitive world. But if it can, then it's again very profound, because then that root, it's red actually, so we'll keep it going, then that root can grow down to depth. Okay? Because it's got enough of this mycorrhizal, water and nutrient to supplement that. And it's all been helped by the biostimulants, initially to get the biochemical water, and then this unavailable water, unavailable nutrient. So now it's got a root, and just say that's to 40 centimeters. And that's before there's actually really much growth on the plant. And once it's got that root system, it's amazing, then it can start putting out its first leaf.
Walter, do you mean millimeters?
No, centimeters. Oh, no, we're talking about centimeter, all right? This is because it's got to survive. Okay? So then, exactly, at this stage, the root-shoot ratio can be 100 to 1. Okay? Because roots are everything.
Here, the mycorrhiza is not taking anything from the tree. It is just providing nutrients and H2O to the tree.
Mycorrhiza isn't taking nutrients from the seed. all that nutrient is going into the root. The mycorrhiza takes some exudates, sugars. I mean, yes, I suppose they're getting some sugars, but no, really initially it's colonizing the roots and it's getting most of its nutrients from the soil surface, the mineral nutrients. It'll get some sugars, but not nutrients from mineral nutrients, right? All the mineral nutrients are actually going to build root because this plant has to grow roots.
It will take some days for the leaves to start functioning, right?
Yes.
Circulation starts?
Yeah, so this might be 40 centimeters and this might be day 6 to 10, right? And you'll see that when you go there and you sow the plant and you go back 1 week later, 7 days later, and you'll see there'll already be roots growing, but you won't have seen a shoot above the ground. But from about day 8, 9, you might just start seeing some shoots. Okay? Once it starts fixing carbon, then of course it accelerates. But the point is it has established this good root system. Once it gets down this deep, then it's actually able to start getting some of this available soil water, right? Because there'll be deeper water in the soil. See, mostly the soil will be very dry at the surface, but down a foot you would be getting perhaps 10% moisture content. There's about 3% moisture content or less up here, right? But it's a very, very tough environment. Okay, once the shoot grows, this is then valuable because then it is able to sort of basically say, can I start harvesting nutrients from the air? Can I start getting hazes and dew? Okay, and Walter more or less sort of said it, like you even have little shoots like that, 10, 15 centimeters tall, and early in the morning, as Walter told us, they're already getting wet. Okay, so very, very quickly, once you've got this root system, then this shoot starts harvesting water from the air. And so then progressively, once you've got the mycorrhizae, the nutrients coming, you've got those supporting the shoot growth, then you're starting to get water harvesting in this plant. Now, the crop that we saw, the really interesting leading crop, we were standing in it and basically the figures were, 40, it was sown in June, May, end of May. May. It got 40 millimeters in July and it got another 60 millimeters in September. Okay? And basically that's the sort of story. So May, June, July, so there was 5 weeks here. August, September, so there's probably another 5 weeks there. But it doesn't matter, right? The fact is, for the first 5 weeks, this plant had to be working on this strategy and had to be harvesting that. Then, of course, with that rain, it was able to grow. But when we saw it, obviously, halfway through November, it was 2 meters tall. There was 10 to 15 tons of biomass. Okay? And we can simply work out if you've got 10 to 15 tons above ground biomass, each ton required about 1,000 tons of water to grow. So we had 15,000 tons of water that crop needed, right? But basically we've only had 100 millimeters of rain. And we know that there's 10,000 square meters in a hectare. So we know that we've had about 1,000 tons of rain. So what are we missing? There's been 14,000 tons or thereabouts of water that's come from somewhere. Now, some of that has come from these surfaces, no question. I don't know how much, perhaps 2,000-3,000 tons, but easily 10,000 tons of water has come from here over those 10 weeks. You see? Because it's a bit like Sherlock Holmes. It's elementary. There can't be any other way.
Could you just say those numbers one more time?
Well, okay, and I mean, I haven't measured it, so all I'm just doing, not hypothetically, it's real. I mean, I can't escape it. But basically, if you have 15 tons of biomass, 2 meters high, and we know biochemically to produce a gram of plant matter takes about 1,000 mils, 1 litre of water. Okay, it takes one litre of water to produce each gram of biomass, just biochemically photosynthesis. So we know that this is basically the water demand to grow that crop. And what we're saying is the rain that we've had over that first 11, 12-week growing season has been this number, roughly. And there has to be water from somewhere. You know, it's not magic, it has to come. So what we're saying, the deficit has to come substantially from this non-available water through the mycorrhizae, plus water from the hazes. But the important message isn't the quantity. The important message, and this is really for Paulie, the important message for Paulie that we have got a sequence of strategies starting off with your zero budget natural farming biostimulant and pre-monsoon sowing strategy. And if we go through this gamble and sequence, you have pulled it off. And that is world significant, because then you have the potential for 365 days green in otherwise arid conditions. So we need to know more, and we need to do studies. And that's why we talk about the little bits of blotting paper. Because if we hang blotting paper up here, we can easily measure how much that is. And we can answer Didi's question, where is that 14,000 coming from? So what we've got here is not a proof, but we have an analysis. You have a reality, there's no question. I was standing in that crop, it's real. We all stood it. I mean, I don't know if you've all been there, but we know that's real. So we can't escape that. But we, like a detective, can look at it and say, well, what's going on? How do we learn from that? And if we can learn, is this something really significant from zero budget natural farming that is significant globally to growing and rehydrating arid zones to get 365 green? Now we can ask these different questions. As you would in irrigated fertilized plants, are much more wasteful. But again, it's a valid point. You know, this is plus or minus. You know, could this have been done with 10,000 tons? Possibly. But doesn't matter. It's still way below the rainfall, right?
What happens in the situation like this?
Yep.
What will happen during the peak summer?
Right. Yeah, good.
When relative humidity is less, temperature is high.
Right.
Soil moisture is also very low. What will happen?
Right. Okay. Very, very significant question.
What are we going to start translating?
We haven't translated anything.
Pardon?
We have
One is biostimulant stimulating the saprophytic water.
I can translate. సో ఇప్పుడు మనం విత్తనం వేస్తానే విత్తనాన్ని బీజామృతంలో ఇది చేసి ట్రీట్ చేసి వేస్తాం భూమిలో చేసినప్పుడు మనకి ఫస్ట్ ఏమవుతుంది సాప్రోఫైటిక్ ఫంగై తినే మనము పూజ లాంటివి వచ్చి వాటిని అటాక్ చేసి తర్వాత అవి కొంచెము ఈ పెరుగుతుంది అన్నమాట జీవక్రియలు ఉత్ప్రేరపరుస్తుంది దాంట్లో ఈ అప్పుడు నీళ్ళు అనేవి ఉండవు కదా నీళ్ళు మధ్యలో అంతా ఎండిపోయి ఫోర్స్ మధ్యలో ఉండే రంధ్రాలన్నింటిలో ఎండిపోయి అక్కడ ఇందాక చెప్పిన మనకి బాటిల్లో చెప్పారు కదా బాటిల్ ఇక్కడ మాత్రం కొన్ని అక్కడక్కడ మాత్రం ఉంది అట్లే పార్టికల్స్ మన కణాల మధ్యలో అక్కడక్కడ వాటర్ ఉంటుంది ఆ వాటర్ని ఈ సాప్రోఫైటిక్ ఫంగై తీసుకొని అది గ్రో చేయడానికి ఉపయోగపడుతుంది పెరగడానికి ఉపయోగపడుతుంది తర్వాత
Water.
సారీ
Yeah.
ఈ సాప్రోఫైటిక్ ఏదైతే ఉన్నాయో దాన్ని పరాన్న బుక్కులు అంటామని తెలుగులో పరాన్న
First little root.
So in process number 1, what in the biostimulant in the Bijamrutam, in the seed coating,
Yes.
do we assume is creating this process of stimulating the saprophytic fungi?
No, it
Is
is.
it sugar in the jaggery? Is it the microbes in the coating?
No, it is stimulating, switching on the activity of natural saprophytic fungi already in the soil. It is making them more active. The things that make it more active is a mixture of amino acids and complex biochemicals from the enteric, that's the digestive system of the animals, both in urine and feces. Now, if you want to go through the biochemistry, but I'd lose you. It's, the various butyric acids and propionic acids and what have you. Very complex.
So it's not the sugars that...
No, there's no sugars in excrement, no.
No, but in the coating, they have
Oh.
sugars, right? You have
Okay, look. No, it's a bit of a red herring at this moment. What are the active ingredients that stimulate the saprophytes? It is, yes, it is those biostimulants from the guts. But no, if you just put sugar on the soil, you wouldn't get the same stimulation of the saprophytes. You'd get some sugar breaking down, but nothing like is needed to drive this forward.
But in the process too, there is the harnessing of water takes place. Process 2.
From 2 it is actually using this water to swell the seed, to germinate that, right? But at the same time, because the biostimulant, process 2 is also switching on all these mycorrhizae fungal. So first it's switching on the saprophytes, then it's switching those on, and because of that biostimulants, it's enabling this process and that process to get started by the mycorrhizae. Now then we had a lot of questions about mycorrhizae A or B or whatever, and yes, there's a whole lot of variation, but actually Suzanne can answer those questions as we get into detail.
And the third,
translation.
సో ఇప్పుడు ప్రాసెస్ 2 అనేసరికి ఆ వాటర్ ఏదైతే బయోకెమికల్ వాటర్ యూజ్ చేసుకొని ఆ విత్తనం పొంగి అది జెర్మినేట్ అవుతుంది. జెర్మినేట్ అయిన తర్వాత ఆ వేర్లకి మైక్రోరైజా మన ఫంగై ఉన్నాయి కదా శిలీంధ్రాలన్నీ అవి కూడా స్విచ్ ఆన్ అవుతాయి. అంటే అవి కూడా ప్రేరేపితమై అవి కూడా వేర్ల నుంచి స్ప్రెడ్ అయ్యి దానికి అంటుకొని ఈ వాటర్ బాల్ చూపించినాం కదా అక్కడ ఈ కణాల్లో ఉండే అక్కడక్కడ లైట్ వాటర్ని అది ఉపయోగించుకుని తర్వాత బాగా మొక్క పెరగడానికి దోహదపడతాయి. దాంట్లో న్యూట్రియంట్స్ కూడా తీసుకుంటుంది అన్నమాట. ఆ చుట్టుపక్కల సర్ఫేస్ ఏరియా న్యూట్రియంట్స్ అక్కడ మనకు ఎండాకాలంలో అది వర్షం ఉండదు కాబట్టి ఆ మధ్యలో ఉండే వాటర్ దాన్ని సాయిల్ పార్టికల్స్ మధ్యలో ఉండే అవైలబుల్ వాటర్ ఉండదు. సో ఏదైతే మనం ఇందాక చూసామో ఈ చిన్న చిన్న వాటర్ దాన్ని తీసుకొని అది గ్రో అవుతుంది.
Yeah, you probably already answered in that. See, so 2 I had germination to here. 3 was this whole thing, process 3, but you may have already, I mean, like there's no hard boundaries, but that's the next step, right? You probably talked about that from what I gather.
And the 4 is
No, the 4th one is because of this nutrient and water, the root is able to go from there down to there. So this is process 4. Okay, so now it's able to get water, including some deeper soil water from the in-soil reservoirs that are still down there. Okay, so that's process 4. And once you've got enough there, then you get up here process 5, which is a shoot. Okay? And then process 6 becomes the capacity of that shoot, as Walter told us, to start harvesting water from the air.
So, can you please elaborate on 5?
On 5, 4, hang on, 5, shoot. Yeah, it's only when that plant has got enough water and nutrients both from here and there that it can start putting the shoot. Remember we said it's really established a root shoot ratio of 100 to 1, and you will find that your basically pre-monsoon seedlings will be very slow in coming up because they will be waiting till they have a root down there. If you fertilize that plant, you would have auxin or growth hormone that would put shoots up, less root, and it would be dead within the next 2 weeks.
Yeah. Process 3M చెప్పాం కదా, మట్టి కణాల చుట్టూ ఉండే వాటర్ని తీసుకొని అది వేరు వ్యవస్థ బాగా డీప్ సైకిల్స్ పెట్టుకుంది. పెరిగి ప్రాసెస్ 4 ఏంటంటే అది పెరిగిన తర్వాత వేరు ఆ లోపల ఉండే వాటర్ చాలా లోతుగా ఉండే నీటిని తీసుకొని బాగా పెరుగుతుంది. పెరిగిన తర్వాత ప్రాసెస్ 5 వస్తాము. ప్రాసెస్ 5 అంటే ఏంటి, అది బాగా పైన ఆకులను వేస్తుంది. ఆకులు అక్కడ నీళ్ళు ఎక్కడ వస్తే ఆకులు వేసేదానికి కింద డీప్ లోతుగా ఉండే నీళ్ళని తీసుకొని ఆకులు వస్తున్నాను. ఇంకోటి ఏంటంటే ప్రాసెస్ 6 ఏంటంటే ఆ వచ్చిన ఆకులు గాలిలో ఉండే తేమని గ్రహించి అవి ఎట్లా కేజెస్ అవన్నీ తీసుకొని పెరుగుతాయన్నమాట. ఇట్లా 5 ప్రాసెస్ మనకు ఉన్నాయి. బట్ మనకి ఆయన చెప్తున్నది ఇంకోటి ఏంటంటే ఈ మనం ఏదైతే ప్రీ మాన్సూన్ డ్రై సోయింగ్ 365 డేస్ గ్రీన్ క్రాప్ విత్తనం వేస్తానే మొలకెత్తడానికి చాలా రోజులు పడుతుంది. సో అది ఎందుకంటే ఈ ప్రాసెస్ అనేది జరిగేసరికి అది వెయిట్ చేస్తూ ఉంటుంది లోపల అన్ని ఇదంతా జరగడానికి. That's very true. I observed in 365 days. It takes a lot of time for germination and then.
Yeah. Process 7 is the fact that you've got reality, you've got that crop. Now, because we're detective, systems analysis, this is reality. It's nothing hypothetical. It's solid. What's hypothetical, not hypothetical, where we still have to work out how did that happen? What is it, the process steps, but we don't deny reality. You're standing on it. Okay, and process 8 is zero budget natural farming, having done something that is unique, which is arid zone, 365 days pre-monsoon, that is fundamental for the future of humanity and food security.
So, process 7 in ప్రతి కిలో 1 kilogram 1 gram of growth కి 1 liter of water అవసరం పడుతుంది. అట్లాగే మార్చిలో నుంచి సెప్టెంబర్ వరకు పెరగడానికి 15,000 లీటర్లు వాటర్ కావాల్సి వస్తుంది. మనకి మాన్సూన్ వచ్చిన తర్వాత వర్షం పడితే మహా అంటే 1000 లీటర్లు పడతాయి. ఈ 1000 లీటర్లని ఇంకా మిగతా 14,000 లీటర్లు ఎక్కడి నుంచి వస్తున్నాయి అది ఆలోచించారు. సో సంథింగ్ సమ్వేర్ ఇట్ ఇస్ కమింగ్. అదేంటి మహా 10,000 లీటర్లు మాత్రం గాలిలో నుంచి తేమ నుంచి గ్రహించగలిగే శక్తి ఉంది. మిగతా 4 లీటర్లు ఈ నీళ్ళలో నుంచి ఈ వీటి నుంచి కణాల నుంచి వస్తుంది. బయోకెమికల్ వాటర్ ఒక 1000 లీటర్లు 2,000 ఉన్నా కానీ ఇవన్నీ కలిపితే ఈ ప్రీ మాన్సూన్ డ్రోస్ అయ్యి ఇయర్ లాంగ్ గ్రీన్ కవర్ వస్తుంది. అది జడ్బీ అన్నట్లు
And when we have those 8 processes, you have done. But now, Susan, can you ask all the hard questions? Because once we have this analysis, we accept that there's 20% that we don't know, but let's look at the questions, because I think when we have this context, then we can answer, it answers a lot of the questions, but I can't read it.
One second. The water that is attached to...
This is still pure chemical water, it's just bound physically, whereas most hygroscopic water has solutes in it, you know, that are chemically attractive. But look, that's a technical oh, go on. It's very precise technical. But look, Susanna, and we'll come to that. Would you like to ask the questions?
I will ask the questions and then I will ask translation every time, if possible. So, during peak summer conditions, did we... This is...
That the one you are asking?
Yes.
Hang on. Yes. One at a time.
Are you requesting questions from...
Yeah. So basically, yes, the whole thing, this is peak summer conditions. We have not had any rain. This is reality. This is happening without rain. Peak summer. Yes.
సో ఎండాకాలంలో అసలు వర్షమే లేని టైంలో ఇదే జరుగుతుంది అనమాట. ఇలా జరగడం వల్ల ఏదైతే ఫస్ట్ సెకండ్ థర్డ్ సైకిల్ జరుగుతున్నాయో దానివల్లనే వేరు వ్యవస్థ తర్వాత మొలకెత్తడం జరుగుతుంది అనమాట. అదే పీక్ సమ్మర్లో జరుగుతుంది.
Okay. Then next question. When microbes woke up, if it's a monocrop, will they feed on monocrop roots?
No, when the microbes wake up, the first microbes are these saprophytes. There are no growing roots at that stage. They are just organic matter that is turned into this water. And then basically when the mycorrhizae come up, it's not a monocrop either. It depends, because if you've got a mixed cover crop, millet and cowpea and, you know, Ricinus communis, you'll have a whole lot of different roots and it's not a monocrop.
సో మోనోక్రాప్ వచ్చిన వెంటనే ఆ వేర్లను తినలేదన్నమాట. ఎందుకంటే సాక్రోఫైట్స్ ఏవైతే ఫస్ట్ మనం మేల్కొల్పే మైక్రోఆర్గానిజమ్స్ ఉంటాయో అవి వేర్లను తినమన్నమాట.
The difference between ectomycorrhiza and endomycorrhiza?
Okay, this is a technical question. What's the difference between an ectomycorrhizae? We can draw that very simply. Here is a root of a plant, and we'll do it cut it in half. An ectomycorrhiza basically has hyphal networks around the outside, a matting, a coating of hyphae. This is called a Hartig net. And basically it'll grow massive hyphae out into the soil from there, and it will grow in between the cells of the root, right? It'll just grow into the intercellular spaces, but it actually won't invade the cells of the root. An endomycorrhizae is more evolved and more sophisticated, and it basically doesn't have a Hartig net, but it will actually colonize a hostorium and a buscule inside the cell, and then it will create the 25,000 kilometers per meter cube of healthy soils of fungal hyphae proliferating through the soils.
Just to simplify, ecto means outside.
And
Are these associations question there for all crops because some crops are non-mycorrhizal. So should we select the crops that have the
Okay. 98% of plants in the world are mycorrhizal, about 20% ecto, 80% endo. There are some plants, the cruciferae, the chenopods, that aren't mycorrhizal. Some of the Banksias have another different sort of mycorrhizal or another microbial symbiosis. So there's a lot of diversity. Okay, but basically 98% of plants are mycorrhizal. The chenopods, the brassicas, they operate on a different system where they exude a lot of acids from their roots, and those acids are helpful in solubilizing nutrients, and they've got fine roots that help them take up a bit more water, but they're nowhere near as competitive in arid conditions like this because these things give them real resilience, competitiveness in very, very extreme environments.
సో ఇందాక అడిగిన క్వశ్చన్ ఒకటి ఎక్కడైతే ఈ మైక్రోరైజల్ అన్ని చెట్లకు అన్ని రకాల చెట్లకు ఈ మైక్రోరైజల్ ఫంగి అనేది అటాచ్ అయ్యి ఉంటుందా కొన్నిటి కాకుండా ఉంటాయా అని సో ఏమంటే 98% ఆఫ్ ప్లాంట్స్కి ఈ మైక్రోరైజల్ అటాచ్ ఉంటుంది అనమాట. సో దానివల్ల ఈ ప్రక్రియ మొత్తం జరుగుతుంది. మిగతా 2% వాటికి వేరే రకంగా జరుగుతుంది. ఇటువంటి మైక్రోరైజల్ ఉండకపోయినా ఆ వేర్లే డైరెక్ట్గా కొన్ని యాసిడ్స్ రిలీజ్ చేస్తాయి. ఆ యాసిడ్స్ ఏం చేస్తాయంటే ఇక్కడ ప్రాసెస్ నెంబర్ 3లో చెప్పుకున్నట్ల ఆ ప్రాసెస్ నెంబర్ 3 ఫంగల్ హైపే చేయకపోయినా మన రూట్స్ అనేవి ఎక్స్ట్రాక్ట్ చేసే యాసిడ్స్ వల్ల అవి తీసుకుంటాయి అన్నమాట. వాటర్ని కావచ్చు, న్యూట్రియంట్స్ కావచ్చు.
Okay.
What are mycorrhizae? Hey, we have answered that question. Mycorrhizae are these symbioses, these marriages between fungi and plants that have a mutual beneficial relationship. The fungi get their sugars from the plant because the fungi can't produce sugars. The mycorrhizae provide the plant with essential nutrients and water and enable plants to colonize extreme habitats. This would not be possible without these fungi.
సో మైక్రోరైజా అంటున్నాం కదా, అప్పటి నుంచి మైక్రోరైజా అంటే ఏంటంటే ఈ ఫంగల్ హైప్ ఏదైతే శిలీంధ్రాలకు ఉన్న పోగులకి మరియు మన వేరు వ్యవస్థకి ఉన్న బంధం అనమాట. రెండు కలిసి ఉంటాయి. సో వేరు వ్యవస్థకి పోషకాలు తీసుకోవాలన్నా, నీరు తీసుకోవాలన్నా, అంటే అందుబాటులో లేని నీరు, అందుబాటులో లేని పోషకాలు తీసుకోవడానికి ఈ హైపీ హెల్ప్ చేస్తుంది. శిలీంధ్రాలకు ఉన్న పోగులు తర్వాత శిలీంధ్రాలకు సొంతంగా ఆహారాన్ని తయారు చేసుకోలేవు కాబట్టి వేల నుంచి ఆహారం తీసుకుంటారు అనమాట. సో ఈ విధంగా రెండు కలిసి ఉండడం మైక్రోరైజా.
So the mycorrhizal always, beneficial to plants? And the answer is no, not at all. They're beneficial to ecology, but when a plant gets stressed and is no longer competitive, the mycorrhiza have them for lunch. You see? Because they basically say, look, this is a stressed, weakened plant, and its space is better used by a healthy plant that is regrowing, and I can use the nutrients in this plant. So I did my work initially back in the 70s on mycorrhizae in forests, and when the plants got stressed, the mycorrhizae became pathogenic and very, very quickly killed the plants with phytotoxins, very rapidly, dead. And basically killed those plants, and then the succession of healthy forests came about through the fungi. What you're saying though is that the fungi drive a lot of that successional ecology. And just as the fungi colonized the soil, we saw in petagenesis, it's that microbiology, the microbial ecology that drives a lot of nature.
So it's very dangerous. Not only takes
అన్ని ఫంగస్ సారీ అన్ని రకాల మైక్రోరైజా అంటే మనం అనుకున్న బంధాలు అంటే వేరు వ్యవస్థకి దానికి ఉన్న బంధం అన్ని మంచివేనా అని అడిగారు అనమాట. ఎటువంటి అంటే కొన్ని దగ్గర శిలీంధ్రాల వల్ల మనం అనుకుంటాం కదా వేరు వ్యవస్థ చనిపోతుంది, చెట్లు చనిపోతున్నాయని. సో అటువంటిది ఏమైనా ఉందా అంటే అన్ని మేలైనవే అంటే అన్ని మేలు చేయకపోవచ్చు అన్ని మైక్రోరైజాలు. బట్ ఎప్పుడు ఈ చెడ్డలు చేస్తాయంటే ఎప్పుడైతే ఒక చెట్టు ఎదగలేకపోయిందో, ఎప్పుడైతే ఒక చెట్టు వాడిపోవడం కానీ అంటే వేరే పరిస్థితుల వల్ల
వాటికి కావాల్సింది
అప్పుడు ఏం చేస్తారంటే ఆ మైక్రోరైజా ఆ చెట్టులో ఉన్న పోషకాలను తీసుకోవడానికి చెట్టుని చంపేస్తారు. చెట్టు కొన్ని విష పదార్థాలు వదిలి చెట్టుని చంపేసి ఒట్టి పెరగలేని లేదంటే దాని చేతగాని చెట్లని.
And because of this, Anne Sophie is going to commission a song that goes something like this. We love you, fungi. Oh, yes, we do. We love you, fungi. Well, you can do the rest. Yeah. She can be on Young Talent Time, right?
The voice.
Next question.
The voice of fungi.
Okay. Of pre-monsoon.
Yeah, okay, now this is
What is the question?
Paulie's question again.
What's the ecological impact of pre-monsoon dry soil?
Yeah, what is the ecological impact of 365 days green pre-monsoon crop production? And in a sense, Paulie's already answered that. It's life and death as climate change intensifies for, 3 billion hectares and 3 to 5 billion people on this planet.
And also, so the question was based on the idea that if you do pre-monsoon sowing, you do 2 months or 1 month earlier, and you don't do any tillage, so you've prepared your soil and the mycorrhiza in the soil. You've absolutely made perfect soil for your monsoon sowing, the next coming crop. So that's
then
a bigger ecological impact.
Beautiful. And we were in the field when we were looking at this, and see these pellets, the seed pellets. What I did again in the 1980s, I put another coating around them, which was often, I had the mycorrhizal inoculum, but then at the end of them, I have another put of coating, and that could be slow release nutrients. And then I put another coating on the outside of that, which was wax, just a thin layer of wax, and I could sow these pellets, and they would stay viable for 2 years, and it's only when they got enough water from rain that they swelled up, the wax was broken, and I really had a time capsule, a seed time capsule, seed microbial rhizobia time capsule to colonize arid degraded lands.
Translation.
And
సో ఇప్పుడు చెప్పిన పాయింట్ చెప్తాను. సో సీడ్ వయబిలిటీ గురించి మాట్లాడుతున్నాం. అంటే జర్మినేషన్ కోసం సీడ్ ఏం చేస్తారని ఎన్ని రోజులకు జరగవచ్చు ఏమంటే మేబీ ఆ పూర అంటే పెలటైజేషన్ అని చెప్తున్నారు మనం, పెలటైజ్ సీడ్ పెల్లెట్స్ చేసుకోవాలని. సో పెలటైజేషన్ చేసిన తర్వాత ఆయన సొంత అనుభవం చెప్తుండ్రు. దాని చుట్టూ వ్యాక్స్ కూడా పెట్టినారంట. అది 2 సంవత్సరాల దాకా ఉన్నాయంట. అట్లనే ఎప్పుడైతే పెద్ద వర్షం పడిందో అప్పుడు మాత్రమే విత్తనం లావు అంటే నీళ్ళని పీల్చుకొని లావుగా అయిన తర్వాత మొలకెత్తింది.
next question.
Continuous rainfall affect these dynamics, for instance, also if we have paddy rice. So if you have continuous rainfall,
Okay.
are these dynamics affected if too much?
Look, if there's continuous rainfall, then the same processes work, but in a sense you're getting germination straight away, you're getting access to water, and in a sense it's a lot easier. Okay, so they're not as dependent on each of these stages, no. And this stage, it's 6 grams of water, but compared to rainfall it's nothing, right? But it's everything in an arid zone to get plants started and survive. So it's short-circuited and accelerated with plenty of rain. Yes?
Translation?
I want to ask a supplementary question for that. If water stagnation happens in that area,
Yeah.
stagnation,
When there's water,
when the heavy rain comes down,
yeah, and it's water saturated and what have you. Well, look, Once you get anaerobic, saturated, waterlogged conditions, yes, these fungi basically don't do very well. I mean, they don't die. Well, they die, but they'll regenerate. But basically, what happens if the water table comes up, then you kill a lot of the fungi, you kill a lot of the roots, and you've got very poor growth, and you are inviting a whole lot of fungal diseases, your phytophthora, your fusariums, your verticilliums, fungal root rots, and yeah, so you've got a big disease problem. Why? Because you haven't got a soil carbon sponge, you've got a concreted catchment without the aerations to give you the sponge. Yeah.
ఎక్కువ వర్షం వచ్చినప్పుడు లేదంటే నీళ్ళు నిలిచిపోయినప్పుడు ఏ విధంగా జరుగుతుంది యాక్టివిటీ అంతా అని అడిగారు అనమాట. సో దానికి ఏం చెప్తున్నారంటే ఎక్కువ నీళ్ళు వచ్చినప్పుడు మనకి ఈ యాక్టివిటీ వల్ల ఎక్కువ యూజ్ ఉండదు అనమాట. డైరెక్ట్గానే చెట్టు ఈజీగా పెరిగిపోతుంది అనమాట. మొలకెత్తడం కానీ, వేరు వ్యవస్థ బలపడడం కానీ, చెట్లు పెద్దవడం, ఆటోమేటిక్గా నీళ్ళు ఎక్కువ ఉంటాయి కాబట్టి మనకు దీనికంటే అది ఎక్కువ వర్త్ ఉంటుంది కాబట్టి నీళ్ళు ఎక్కువ తీసుకొని పెరుగుతాయి. బట్ స్టాగ్నేట్ ఇప్పుడు వాటర్ ఎప్పుడైతే సాచురేషన్ మించిపోయి స్టాగ్నేట్ అయితే అప్పుడు ఏమవుతుందంటే ఫంగి అన్ని చనిపోతాయి అనమాట. మనకు యూజ్ చేసే ఫంగి అన్ని నీళ్ళలో చనిపోతాయి. చనిపోవడం వల్లనే డిసీజెస్ వస్తున్నాయి అనమాట మనకు చెట్లకు. సో దీనివల్ల అర్థమయ్యేది ఏంటంటే సాయిల్ అనేది స్పాంజ్లా ఎప్పుడైతే ఆర్గానిక్ కార్బన్ పెరిగి స్పాంజ్లా ఉందంటే స్టాగ్నేషన్ ఉండదు అనమాట, వాటర్ సర్క్యులేషన్.
స్టాగ్నేషన్ అయినప్పుడు అవన్నీ చచ్చిపోయి.
Next question. There are some fungi that eat the roots. Are they affected by inoculants?
Right. There are pathogenic fungi that kill plants. Okay? And they are affected both by the biostimulant but also by these mycorrhizae. It is like a war zone in the soil. There's 10 times more microbial life underground than there is life above ground. And they're all competing. But what happens is that basically if you have a mycorrhizal plant, all the nutrients are being taken up by these mycorrhizae, and basically they are preventing disease organisms being able to attack that plant. Okay, so they both create an immunological response. So because of this mycorrhizal infection, these roots have got chemical compounds, we call them phytoalexins, very complex immunological, and that actually protects the roots. They also increase their exudation, and they then support a very, very active rhizosphere. That's spelled R-H-I. A rhizosphere, which is a zone of very, very active bacterial and actinomycete activity around the root, which is effectively a protective zone, if you like, a force field that avoids things like phytophthora or fusarium or verticillium coming in to attack the root. But what happens if the plant is stressed? Then it can't have the exudates, the rhizosphere weakens, and then the plant becomes more vulnerable. So there are very complex, elegant, sophisticated relationships. Healthy plants with good exudates are totally immune, but once the plant gets stressed, it's vulnerable. But a fertilized plant, a heavily fertilized plant where you don't have these mycorrhizae, and you're giving excess leakage of nutrients, nitrates and stuff, will often induce diseases on those roots, and then you're in an endless cycle of biocides and death.
ఈ ప్రాసెస్లో మనకు మైల్ చేసే దాని గురించి ఇంతవరకు మాట్లాడుకున్నాం. సో చెడు చేసేవి ఏమైనా ఉన్నాయి, ప్లాంట్స్కి చెడు చేసే మైక్రోరైజా ప్లాంట్స్ చెడు చేసే ఫంగై ఏమైనా డెవలప్ అయ్యి మన వేరు వ్యవస్థను నాశనం చేసే అవకాశం ఉన్నాయని అడిగినారు. సో దానికి ఏం చెప్తున్నారంటే ఉన్నాయి, అవి కూడా పెరుగుతూ ఉంటాయి. ఎలా అయితే మంచి ఎంత ఫాస్ట్గా పెరుగుతుంటే చెడు కూడా అదే విధంగానే పెరుగుతుంటాయి. కానీ ఏమవుతుందంటే ఈ రూట్స్కి మంచి అంటే మన మైక్రోరైజా ఏవైతే మనకు హెల్ప్ చేస్తున్నాయో అవి ఎక్కువగా అటాచ్ అయిపోయి ఎండో మైక్రోరైజా ఏమైతే అంటే ఎక్కువగా డెవలప్ అయిపోయి వాటి మధ్య సంబంధం ఆల్రెడీ స్టార్ట్ అయి ఉంటుంది అనమాట. సో అవే ఒక నెట్వర్క్ లాగా ఫార్మ్ అయిపోయింటాయి ఫస్ట్. ఇంకా ఒకవేళ అటువంటి ఏమైనా హాని చేసే ఫంగై ఏమైనా పెరుగుతుంది అని చెట్టు కనిపించినట్లయితే అది సడన్గా ఏం చేస్తుంటే ఎక్కువ సంఖ్యలో పదార్థాలని పిండి పదార్థాలను రిలీజ్ చేస్తుంది అనమాట వేరు వ్యవస్థ దగ్గర. ఒక రైజోస్ అదే వేరు వ్యవస్థని షీల్డ్ లాగా తయారు చేస్తుంది. అదే ఒక షీల్డ్ లాగా ఫామ్ చేసుకొని అదేది లోపలికి రాకుండా కాపాడుకుంటుంది అనమాట.
అంటే ఇంకోటి కూడా చెప్పారు, కెమికల్ దాంట్లో ఫెర్టిలైజర్స్ వేసిన దాంట్లో ఈ రైజోస్పియర్ మైక్రోరైజా ఎక్కువ ఉండదు కాబట్టి ఈ ఎక్కువ ఫెర్టిలైజర్స్ అన్ని నైట్రేట్స్ రిలీజ్ చేస్తూ ఉంటాయి. ఆ నైట్రేట్స్ కోసం ఈ అటాక్ ఎక్కువ అయిపోతుంది వీటి నుంచి, వెట్రిసైలియము ఇవన్నీ. సో ఆ రైజోస్పియర్ అనేది దాంట్లో ఉండదు అనమాట కెమికల్ ఫార్మింగ్.
స్ట్రెస్ ఇంకోటి చెప్పారు సార్, స్ట్రెస్ ఎక్కువ అయినప్పుడు ఇటువంటి రైజోస్పియర్ స్ట్రెస్ ఎక్కువైన ప్లాంట్స్కి ఎక్కువ రోగాలు ఎందుకు వస్తాయంటే స్ట్రెస్ తట్టుకోలేనప్పుడు రైజోస్పియర్ని డెవలప్ చేసుకోలేము అనమాట స్టీల్. అందుకనే ఫంగస్ ఎక్కువ అంటే విషం లైక్ హాని చేసే ఫంగస్ ఎక్కువగా డెవలప్ అయిపోయి ఆ చెట్టుని చంపేస్తుంది.
How does tillage affect pre-monsoon dry season?
Right. How does tillage of the soil affect this capacity to establish plants? Well, generally speaking, it's deleterious because first of all, you're breaking up the soil and if anything, increasing evaporation. So any water that was left in the soil, okay, would be now evaporating and getting, even this water is getting less and less because it's evaporating. Okay, now obviously if it's if there's no plants growing there, there's no mycorrhizal fungi. But if you tilled when there's mycorrhizal, you again break up all the fungi and you do all that destruction. So generally speaking, there's no need for it because, like pre-monsoon sowing, you're sowing onto red soil surfaces, so you're not benefiting it by having tillage. In fact, there's really no need for tillage anywhere in agriculture. We generally till to try and get rid of weeds, but we can manage weeds much better through competing them rather than trying to kill them.
PMDS చేసే ప్లాట్స్ లో మనం దుక్కి దున్ని చేసుకుంటే టిలేజ్ దుక్కి దున్ని చేసుకుంటే నష్టం ఏముంటుంది అంటున్నారు. సో ఏదైతే మనం ప్రాసెస్ 1 అని చెప్పుకున్నారా, సో ఆ ప్రాసెస్ 1లో రిలీజ్ అయిన తేమ ఏదైతే ఉందో అదే మనం తక్కువ అనుకుంటున్నాం, మొలకెత్తడానికి సరిపోతుంది అని. సో ఆ వాటర్ కూడా ఎవాపరేట్ అయిపోతుంది, ఆవిరి అయిపోతున్నాయి అనమాట ఎండ వల్ల, ఆ దున్నుకోవడం వల్ల. సో ఆ వాటర్ ఆవిరి అయిపోవడం వల్ల మనకు జర్మినేషన్ జరగదు. సో ఈ ప్రాసెస్ అంతా జరుగుతుంది సార్, ఫస్ట్లోనే మనం ఫ్లాప్ అవుతున్నాం కాబట్టి మిగతా అంతా ఆగిపోతుంది అనమాట. సో అసలు పిఎండిఎస్కే కాదు,
ఏమన్నంటే ఇప్పుడు మనం భూమిని దుక్కి వేయడం వల్ల ఇప్పుడు ప్రాసెస్ ఏ ఉంది ఇక్కడ ఫస్ట్లో విత్తనం చుట్టూ మొలకెత్తడము, దానికి ఏమి బయోకెమికల్ వాటర్ కావాలి. ఆ బయోకెమికల్ వాటర్ అంతా ఆపరేట్ అయిపోతుంది. తర్వాత ఈ కణాల మధ్య మట్టి కణాల మధ్యలో ఉండే వాటర్ అంతా అది మాయిశ్చర్ కూడా అది ఆపరేట్ అయిపోతుంది. సో ఆ ప్రాసెస్ నుంచి ఈ మన సాప్రోఫైట్స్ రావడం కూడా తగ్గిపోతుంది అన్నమాట టిల్ చేయడం. సో విత్తనం అనేది మొలకెత్తడం అనేది చాలా కష్టం అవుతుంది. సో ఎక్కడ కానీ మనకి ఈ టిల్లేజ్ చేయాల్సిన అవసరం లేదు వ్యవసాయానికి, అది దానివల్ల నష్టమే కానీ ఫలితం ఉండదు.
Alright. Next is the diversity of crop. How does that affect the mycorrhizae?
Right. The diversity of crop, how does it affect the mycorrhizae? It's beneficial because, in a sense, the more different diversity of plants you're growing there, you've got roots at different depth. Each plant has its own mycorrhizae associated with it, but the whole use of that soil profile is much, much more effectively colonized by roots and fungi when you have multiple species. The fungi are actually very promiscuous. So they will, in a sense, join up with other plants. So you'll have, different grasses or even grasses to monocot, fungal grasses to dicot, fungal connections. So you almost end up with a network, colony, soil microbial interface across that whole system. And you might have read some books, you end up with these mycelial mats in soil stretching hectares in scale that all communicate with each other through these fungal connections. But yes, there's no real deleterious effect. Even if you put a brassica in as some of it, it would just leave a little bit of isolated space with relatively less mycorrhizae, but it just keeps on colonizing what it wants.
సో పిఎండిఎస్లో మనం వివిధ రకాల పంట అంటే వైవిధ్యం ఎక్కువ ఉన్నంత మైక్రోరైజర్ పని ఏ విధంగా ఉంటుంది అంటే అది మంచిగా చేస్తుంది అనమాట. సో ఎన్ని రకాలైన ముక్కలు వేసామంటే ఒకవేళ ఏకదళ బీజం వేస్తే ఒక రకమైన వేరు వ్యవస్థ ఉంటుంది, ద్విదళం అయితే ఇంకో రకం లోతుగా ఉంటాయి అనమాట. సో ఈ వేరు వ్యవస్థ ఏదైతే వేరు వేరు దశల్లో అంటే కొంచెం లోతుగా పోయేవి కొన్ని పైనే ఉన్నా ఉంటే ఈ ఫంగల్ హైఫై కూడా అంత ఎక్కువగా పనిచేస్తుంది అంటే పెరుగుతాయి అంతా ఒక కొన్ని హెక్టార్ల వరకు కూడా కాసి ఉంటే పెరుగుతాయి అనమాట. మనం అనుకుంటా ఉన్నాం ఒక మొక్క మాట్లాడుకుంటాయి, ఒకదానికొకటి ఇచ్చిపుచ్చుకుంటాయి అంటాం కదా న్యూట్రియంట్స్ అన్ని. సో అవి జరిగేది దీని వల్లనే అనమాట. ఇవే ఒకదాని నుంచి ఒకదానికి మాట్లాడుకోవడం కానీ న్యూట్రియంట్స్ ని సప్లై చేసుకోవడానికి కానీ.
Mycorrhizal highway common highway.
Okay. Then the next, whether there's like a lot of different species and how that's affecting the crops.
Any specific connection to their particular host or something?
Yeah. There's a whole diversity of mycorrhizae in soils. We've identified about 30 different species, but the concept of a species in this promiscuous world is very, very difficult. But also, especially for the endomycorrhizae, very rarely can we actually identify them because you have to produce a spore and only then identify them in the old system from the spore characteristics. And if they didn't produce a spore, you couldn't actually characterize them. Nowadays, with DNA analysis, we can do a lot more, but what happens now, we get this alphabet soup of nucleotides, millions of them, and we don't know what they mean. It's a bit like, having a bowl of alphabet soup and then seeing if the Gettysburg Address comes out of your soup bowl, right? Because it's just so much genetic information, and what does it mean? But that's not, okay, that's just our classical 19th century look at stamp collecting or taxonomy. What we really think about is, look, here is a functional interface. And this is a profound thing for zero budget natural farming, dry season cropping. If you have a healthy soil microbial root interface in that soil, and if you're managing that through the right conditions, don't worry about what species are. They know who they are. They know who they want to eat and colonize and, infect and work with. And all you have to do is create the right environment and enjoy the benefits.
మైక్రోరైజల్ డైవర్సిటీ గురించి అడిగినారు. సో మైక్రోరైజల్ డైవర్సిటీ అవే ఉంటాయి, పెరుగుతూ ఉంటాయి, దాన్ని మనం ఏం చేయనవసరం లేదనమాట. మనం జస్ట్ అవి పెరగడం అంటే ఏ చెట్టుకి ఏ మైక్రోరైజర్ అటాచ్ అవ్వాలి ఏది అనేది అవి వాటికి చూసుకుంటాయి. మనం దాన్ని స్పెసిఫిక్గా తెలుసుకోని అవసరం లేదు. జస్ట్ అవి పెరగడానికి ఒక ఎన్విరాన్మెంట్ని మనం క్రియేట్ చేస్తే సరిపోతుంది. వాటి పని అంతా అవే చేసుకుంటాం.
Local inoculants from local animals versus non-local animals, how to see how they affect mycorrhizae.
Right. This is now coming back to the biostimulant around the seeds, and is somebody's dung better than somebody else's dung, or somebody's urine better than other people's urine? The answer, I mean, we've done some of this. The answer is no, they're pretty much the same, but obviously there's going to be some differences. So, yes, whether camel dung is better in that arid zone than donkey dung, hey, there's a lot of work for you guys to do. But generally speaking, it's more we are switching on, it is, here are these biochemicals from excreta, from the anaerobic gut fermentation product from animals that are acting as biostimulants to these fungi. It's the butyric acids, the propionic acids, it's extremely complex. We don't understand all the active details, but basically you get 95% effectiveness right across the board, but there could be specificities. I mean, for example, in Queensland we have cassowaries. Cassowaries are living dinosaurs. They're big birds, 2 meters high, purple necks, and really beautiful birds, very strong, very dangerous if you fight them. And they basically are living off rainforest fruits. And those seeds will only germinate when they've gone through the gut of a cassowary. We have spent decades trying to germinate these Davidson plums and can't do it. We've got to put it through the intestine of a cassowary and then it germinates. So whether cassowary dung has the unique magic formula, that we will leave that to the young zero budget natural farming microbiologist to work out.
సో లోకల్ అనిమల్స్ గురించి మనం అనుకుంటుంటాం కదా, ఈ జంతువు పేడ మంచిగా ఉంటుంది, ఈ జంతువు నుంచి ఎక్కువ ఉంటుంది అని. సో అది మనమే టెస్ట్ చేయాలి. ఒక్కొక్క ప్లేస్లో ఒక్కొక్క విధంగా జరుగుతుంది. సో ఫర్ ఎగ్జాంపుల్ ఒక జాతి చెట్టు ఒక పక్షి తిన్న తర్వాత దాని ఇంటెస్టైన్, దాని జీర్ణ వ్యవస్థ నుంచి బయటకు వచ్చిన తర్వాత మాత్రమే మొలకెత్తుతుంది అనమాట. సో అట్లా కొన్ని రకాల చెట్లకి కొన్ని యానిమల్స్ యొక్క ఇంటెస్టైన్లో ఉన్న యాసిడ్స్ వల్ల బయోస్టిమ్యులెంట్స్ జరుగుతుంది అనమాట. ప్రాసెస్ నెంబర్ 1 జరుగుతుంది. సో అది కనుక్కోవాల్సిన బాధ్యత మన మీదనే ఉంది.
I can add, in the 1970s, I was working on tropical pasture, bringing legumes into those pastures for minimal input agriculture in Australia. I collected about 300 different strains, isolates of mycorrhizae from all over northern Australia, these dry savanna, rangelands. And basically there was some specificities to different phosphorus types, iron phosphate to rock phosphate, calcium phosphate, etc. There was some specificity to different hosts, but generally speaking, 90%, commonality. So the key issue was actually, are we stimulating this activity, or are natural factors impeding, repressing that activity? So the actual health of the biostimulant was the most important thing. I then went to Nanjing and China to look at what are the factors that stimulate or repress mycorrhizal activities in soil, because I had to go to an agricultural soil which I thought had high mycorrhizae, so I knew what was killing them or what could hurt them. And basically the same story there, the same animal excrement byproducts were the most significant beneficial stimulant factor.
సార్ ఒకసారి ఇదే విధంగానే ఆయన టెస్ట్ చేద్దాం అనుకున్నారంట. అంటే ఏ మైక్రోరైజర్ ఎటువంటి దాటికి
టెస్ట్ చేద్దాం. 1970 సంవత్సరంలో ఆయన పాస్చర్స్లో పనిచేస్తున్నారు, గడ్డి సవరణ అక్కడ పనిచేసి మైక్రోరైజర్ డిఫరెంట్ జాతుల్ని తీసుకొని వచ్చి కలెక్ట్ చేసి వాటిని టెస్ట్ చేస్తున్నప్పుడు అవి విత్తనాలు వాటికి అన్నిటికీ చాలా వరకు మనిషి బయోస్టిమ్యులెంట్సే మెయిన్ ఎక్కువ పనిచేస్తాయి బాగా. అవి అనిమల్ యొక్క పశువులు
Bring an exotic plant that hasn't been at all in that soil, then there are times where we need to bring its mycorrhiza. So, for example, when we brought pinus to Australia, for the first 50 years we couldn't grow them, okay? And it was only when we had the right mycorrhiza that they would grow. Okay, so yes, that applies. We have to bring its inoculum across to make some things grow. Other things are, again, promiscuous. As a general rule, though, when we add inoculated mycorrhiza from elsewhere, it may be 20% more effective, but it mostly doesn't survive. Very rarely does the introduced organism establish itself and become dominant or even be able to be covered within 2-3 years. So mostly it's a native mycorrhizae and its biostimulation that's a key factor rather than bringing in unique inoculum. This is very important because we're going to get a lot of biochemical fertilizer companies selling you proprietary, patented bugs. And they will sell you bottles of jungle juice to pour onto your soil. You're better off pouring dollars on your soil or, rupees or whatever. But the point is, very expensive, inocular, but mostly your native organisms through good soil management and biostimulation will be just as effective and much, much cheaper. And you are into zero budget natural farming, so I wouldn't recommend wasting money on Monsanto.
Supplement to that question, what about the hybrids? Because record down.
Yeah.
What about the hybrids, which is that because the endomycorrhiza is depending on the genetic code, they recognize the genetic code of the particular species because it is species specific, no?
As I said, there are some specificities like the pines, but generally speaking, I mean, the gramineae, the grasses, they will all cross-infect. The legume, I mean, it's generally speaking, 90% again, they're promiscuous and is really creating this soil microbial interface through the biostimulation process rather than I have particular organisms for particular crops.
I'm interested to know about the hybrids.
But hybrid, yeah, look, I mean, no, this is this is microbiology. They are all hybridized. They will all promiscuously interconnect. They will swap genes. They will swap protoplasm. So it is like one soup under there. I mean, it's a network of genetic change. And actually, it's quite fascinating because if you have a toxic soil, you bring an organism or select an organism that's resistant or able to break down that toxin, and it will pass that biochemical activity on to other organisms. So 5 years down the track, many organisms have got the enzymes to break down that toxin. This is actually an important thing in terms of inoculum, for example, and Dedi's in that situation, and we're going there again in February because, see, California, they took these eucalypts from Australia, but they just took the trees. They didn't bring the fungi with them. And so then the litter from those fungi is very acid, and it doesn't break down very well in California because they don't have the fungi to break it down. Yeah, from the eucalypts. So every 3 years Hollywood burns because they have wildfires. Because the eucalypts are producing litter and fuels and fires. And so what we now have the opportunity to say, look, do we have fire or do we have fungi? Can we use the fungi to accelerate the breakdown of the litter, make soil carbon sponge, make more hydrology, reduce fire risks because we reduce the fuel, we've increased soil moisture.
సో ఇంతకుముందు అడిగిన క్వశ్చన్ ఏమంటే ఫస్ట్లో ఒక ఏరియాలో లేకుండా కొత్తగా తీసుకొని వచ్చి ఒకవేళ చెట్లు కానీ మొక్కలు కానీ విత్తనం కానీ విత్తితే ఏ విధంగా ఈ మైక్రోరైజర్ దానికి అసోసియేట్ అవుతాయి మన దగ్గర ఉన్నవి. సో అవి దానికి తగ్గట్టుగా కరెక్ట్గా పనిచేస్తాయా ఇటువంటి ఈ మొత్తం ప్రాసెస్ అంతా కంప్లీట్ అవ్వడానికి అని అడిగినారు అనమాట. సో కష్టం అవుతుంది అంటే దానికి ఎగ్జాంపుల్ కూడా చెప్పారనమాట. పైన చెట్లు ఒక దగ్గర నుంచి తీసుకొని వచ్చి ఇంకో దగ్గర నాటినప్పుడు ఓన్లీ చెట్లు మాత్రమే తీసుకొని వచ్చినా దానికి సంబంధించిన మైక్రోరైజర్ తీసుకొని రాలేదు కాబట్టి అవి పెరగలేదు. ఇంకొకసారి మళ్ళీ ఇనాకులం తీసుకొని వచ్చిన ఆ మైక్రోరైజర్ ఇక్కడ పనిచేయలేదు అనమాట, తీసుకొని వచ్చిన ప్లేస్లో పనిచేయలేదు. ఎందుకంటే ఒక ప్లేస్లో మైక్రోరైజర్ అంటే ఫంగై ప్లస్ ఇనాకులం మనం ఏదైతే జంతువు యొక్క పేడ కానీ మూత్రం కానీ వాడుతున్నాం అది ప్లస్ విత్తనం లేదా చెట్టు ఈ మూడు అసోసియేట్ అయ్యి ఉంటాయన్నమాట. సో ఈ మూడింటిని బేస్ అయ్యి ఈ ప్రాసెస్ అంతా నడుస్తుంది. తర్వాత నెక్స్ట్ క్వశ్చన్ హైబ్రిడ్స్ గురించి అడిగారు. సో హైబ్రిడ్స్లో ఏ విధంగా ఉంటుందంటే హైబ్రిడ్స్ సంబంధించిన విత్తనంలో జర్మ్ప్లాజం కానీ ప్రతి ఒక్కటి డెవలప్ చేయడం కాబట్టి దాంట్లో అది
So actually, I need more clarity about that.
What చెప్పింది చెప్పండి.
I చెప్పింది transparent.
I think he spoke about that. వారు చెప్పింది చెప్పిన తర్వాత మీరు క్లారిటీ అడగండి. వారు చెప్పారు జవాబు ఇచ్చారు. ఫంగై కీప్ చేంజింగ్, మారుతూ ఉంటాయి. That is what Walter has said.
Yeah, and
ఫంగై అనేది ఏదో ఫిక్స్డ్ గా అనేది కాదు. Promiscuous అంటే
we're getting into very exciting, interesting things, but it's real, we've done it. See, if we want to break down toxic soils, chemically toxic soils, we do what we call enrichment cultures. So we start off with low concentrations of toxins, we have fungi that can tolerate them, and then progressively add higher concentrations of toxins. So all the time we are selecting more toxic breakdown capacity. And very quickly, we can get 500% breakdown capacities because the genes, the biochemistry has selected and evolved and improved.
Can we do that for saline soils and other what we now call soils which are degraded?
But saline soils aren't degraded. Saline soils are beautiful. Because the mycorrhizal fungi can take fresh water from saline soils, leaving the sodium and the chloride behind. So the saline player lakes in Rajasthan and Australia are water harvesting surfaces that the mycorrhizal plants around those player lakes can use to grow with fresh water. It's 10 times seawater. But the hyphae growing into that, because they don't take, as Didi was saying, they don't take up the water as if it was a straw. They take it up across a membrane selectively, leave behind the sodium ions, leave behind the chlorine ions, and just move water through ion channels. It's an active process, and of course a bit like what we talked about here, the biochemical water and the water used from these surfaces, they are using the same, here's a little diagram, they are using the same membrane interface as a quality control system taking water, leaving behind the toxic salt. Farming system in saline soils that are very, very productive.
We have seen how microbes and mycorrhizae are helping a plant out
Yeah.
in moisture stress situations like PMDS. And we are also applying biostimulants with foliar sprays. So any comment on philosphere, microbes?
Okay, yes. So philosphere or adding biostimulant to the foliar leaf. And it's again a mixed, like a multiple effect. Obviously, if you're adding animal nitrogen, urea, ammonium ions and what have you, then there's a fair amount of nutrient absorption able through the leaf. Okay, so the plants will green up very rapidly because they've got more, not just nitrogen, but they'll have more sulfur and zinc and stuff from the biostimulant. So there's a fertilizer effect. But also they will stimulate the growth of bacteria and fungi on the leaf surface. Every surface in nature is covered with bacteria and microbes. And normally 99% of the time that's beneficial because like here, you end up creating more immunity. Just as a rhizosphere, you end up with a phyllosphere that protects. But sometimes you can spray your leaves and all of a sudden pathogens, leaf pathogens can become, more aggressive. So, 90% of the time it's benefiting the plant, but sometimes it can actually kill the plant. But then you just have to look at that situation, and where there's a pathogen that could infect the leaf, then you may not want to use that much foliar spray or so much nutrient. Or you put the biostimulant in a form that hasn't got a lot of nutrient with it.
How it helps? What are one thing? Some of our
as we said, look, we looked at this. How does pre-monsoon dry sowing work? What could be the processes involved? We can be here 80% confident that yes, we know this is reality, the yield. Yes, I think we have enough, but there's 20% here, details of, we need to do tests. We need to see what are the best ones. And that's in a sense what zero budget natural farming and your trials and your observations are doing. And if you're collecting that information, progressively you are building a world-class information understanding base that is leading the world, but that solves those problems. I don't have all the precise answers by any means, right? Sorry.
ఇలాంటి స్ప్రే చేస్తున్నాం కదా, సో అది ఎటువంటి ఎఫెక్ట్ ఉంటుంది అని అడుగుతున్నారు. దానివల్ల డైరెక్ట్గా ఆకు మీద నైట్రోజన్ కానీ ఏదైతే పోషకాలు ఉన్నాయో అవన్నీ డైరెక్ట్గా అంది వెంటనే తొందరగా వృద్ధి చెందే అవకాశం అయితే ఉంది. బట్ ఒకవేళ హాని చేసేటువంటి ఫంగై కానీ ఏవైనా ఉంటే ఆకు మీద అవి కూడా తొందరగా డెవలప్ అయిపోయి రోగాలు కానీ తెగులు కానీ సోకే అవకాశం ఉంది. ఫోలియా స్ప్రే చేయండి.
ఆకు మీద నచ్చగానే కాదు, మంచి మేలు చేసే ఈ హైపర్ డెవలప్ అయ్యి ఆకు పైన ఫిలోస్పోర్ లాంటి డెవలప్ అవుతుంది. ఇక్కడ రైజోస్పియర్ డెవలప్ అయిందో అది ప్రొటెక్ట్ చేస్తుంది. తర్వాత పోషకాలు అందజేసి బాగా తొందరగా వచ్చేది.
Last question, I think, was based on the observation from some PMDS that some seeds were germinating and other seeds were not germinating. So we're asking, do we have like a number of different types of crops that is limitation, or why some seeds from some species did not germinate and others did germinate?
Very, very good question, and exactly what you would expect. Because as we said, we've gone through this very fragile sequence of processes, and the first process were these biostimulants and able to start these saprophytes to generate enough biochemical water to germinate that seed. Okay? And that's very much a case in some species there would have been enough water, in other species there may not have been enough water. So the harder, the drier, the more variable different species are more adapted. But that's okay, because when you do get rain, then those other species might germinate later.
The one thing, major thing that we observe is, because some species are germinating well, some species are not germinating well. Maybe the first thing, the mistake they did was they plowed the land. There is very hot summer and lot of evaporation of water, available moisture. And also the second thing is this surface water around this molecules may evaporate.
Yeah.
So that may be one reason. Some species may need more water.
Yeah.
And availability of this saprophytic fungi.
Yeah. But look, there are no mistakes. You are not making a mistake. You are doing exactly what nature does. It produces billions of seeds, broadcasts billions of seeds in mixtures, and then allows the best and most competitive to establish and survive.
So you mean it's good?
So you haven't lost anything. It's a bit like you've given everything the very best chance, and even if it doesn't grow, you've added nutrients to the soil through the seed and the biostimulant. So what I'm saying is you've had a risk minimization strategy by adding multi-species crops, and you're then giving yourself the very best chance that your pre-monsoon season comes up, but there's no negatives because you're not losing anything, but you're learning. So never negatives, just a journey, an adventure, and having fun.
సో కొన్ని రకాల పిఎండిఎస్లో కొన్ని రకాల విత్తనాలు ఎక్కువగా మొలకెత్తుతున్నాయి, కొన్ని విత్తనాలు సరిగా మొలకెత్తట్లేదు అని చూస్తున్నారు. సో ఎందుకు కారణం అంటే మనం చేస్తున్న విత్తన శుద్ధి కానీ మనం అప్లై చేస్తున్న దాంట్లో మనం అనుకుంటున్న ఫస్ట్ ప్రాసెస్లో ఉన్న వాటర్ ఏదైతే సరిపోక ఫస్ట్ ప్రాసెస్లో ఉన్న వాటర్ తేమ ఏదైతే దానికి సరిపోక జర్మినేట్ కాలేకపోవచ్చు. కొన్ని రకాలైన విత్తనాల్లో సరిపోయినటువంటివన్నీ ఆ తేమతోనే మొలకెత్తుతున్నాయి. ఒకవేళ మొలకెత్తలేకపోతే వర్షం పడిన వెంటనే మొలకెత్తుతున్నాయి. ఒకవేళ అసలుగా మొలకెత్తలేకపోయినా మనకేం నష్టం అయితే లేదు. దానివల్ల అది ఆ విత్తనాలు మళ్ళీ డీకంపోజ్ అయిన తర్వాత మన లోపల ఉన్న మైక్రోఆర్గానిజమ్స్ అన్నింటికి ఆహారంగా తయారవుతున్నాయి. సెకండ్ సార్ చెప్పింది దున్నుతున్నారు అంటే మనం ఈ పిఎండిఎస్ చేసేటప్పుడు కూడా ఇంత డెవలప్ చేసిన తర్వాత కూడా మనం దున్ని చేస్తున్నాం. సో దానివల్ల డ్యామేజ్ ఇంకా ఎక్కువ ఉంది. ఈ ఫస్ట్ ప్రాసెస్లో అనుకున్న వాటర్ ఏదైతే ఉందో ఆ తేమ ప్రొడ్యూస్ అవుతుంది, మనం దాన్ని ఆవిరి రూపంలో బయటికి పంపించేస్తున్నాం. సో దానివల్ల కూడా ఎఫెక్ట్ అవుతున్నాయి అన్నమాట, జర్మినేషన్ రాకపోవడానికి కారణం. బట్ ఏదైనా మనం చేస్తుంటేనే మనకు అవగాహన వస్తున్నాయి. ఇవన్నీ చేసి మనకు నెగిటివ్స్ వస్తున్నాయి ఇప్పుడు కాబట్టి మనకు తెలుస్తున్నాయి. ఫెయిల్యూర్ అయితే కాదు, ఇంకా ఎక్కువ చేస్తేనే మనకు తెలుస్తుంది.
The water that is basically used to make the hydrocarbon. So CO2 plus water.
Water. Is that in the seed pellet? Is it part of the seed pellet, or is it in the mulch, or is it in the...
No. It's in the organic matter, the residual organic matter in your soil. Okay, so your soil, and this is why the sponge is so important, because the sponge is stored water. When we make organic matter, when we store carbon in the soil, we are storing sunshine, we are storing carbon, and we are storing water. And we can recover all of those. And so the water comes from that stored, the water that was used to make it is then released again when it's broken down.
Okay. So in the soil, you're saying there's water.
Pellet, well, you mean in the biostimulant coating is basically, as we know, it's dried urine, cow dung, some lime and some soil and some molasses, different combinations. Sorry?
Not in the seed coating. No sugar in the seed. But then we're also applying about 400 kilograms of the soil.
Yeah, but Paulie, please understand. Paulie.
4 times the recommended dose by Palek.
Yeah, but Paulie, the quantity of seed pellet coating that you're putting there is infinitesimal compared to that soil volume, right? So you're 0.1, and so it's not going to drive the biochemical. That comes from your stored organic matter in that soil. The more organic matter you've got, the better your sponge, the more biochemical water reserve.
But just taking Zakir's point, that some seeds didn't germinate.
Yes.
So does it mean we thought that there will be some seeds which need to be pelleted. But what are the lessons coming from seeds which didn't germinate, or it's a trial and error, so we'll have to go through
okay, I think, look, it's
list of seeds which didn't germinate, or it keeps changing.
no,
Can we say,
no, okay, I think we are sometimes too clever as humans wanting to control things. We have put lots of seeds. Just say we put 1 million seeds on the surface, and it's just a matter of luck which seeds fall in a suitable habitat where that biostimulant has access to enough organic matter to generate enough biochemical water for that water to germinate that seed, because there's been no rain. You see, and it's a. Sorry.
I'm thinking that, just thinking aloud, that instead of
10,000 square meters, and if you get one healthy plant per square meter, you've got more than okay. It's wonderful. You see, and so you've got 10,000 successes, but if you put 1 million seeds out, you see, it's okay. There's no failures, right? Because, and even if you only have 5,000 plants, they'll grow bigger and compensate. So, like nature is very, very flexible and accommodating.
But I think the point you're making is where we have sown the seed,
Yeah.
the availability of
The local conditions matters, yes. It is valid. It is valid if you have some mulch and stuff. It probably helps a lot. Or if you've got better conditions, or we find that we can broadcast seed, but then we have a roller to press the seeds into the soil a little bit, and that often helps germination very much. But look, yes, these are all little practice trials and lessons, and very quickly you say, look, I get an extra 40% response from these lessons.
What do we have? One last
Yes.
question.
While going for sowing of a particular crop, should we keep in mind?
Go on, yeah? Yeah, sorry, yeah. Yeah. No, it's okay.
While going for sowing of a particular crop, should we keep in mind the seasonality of a crop or not?
Look, the question is, should we be worried about the seasonality of that crop when we plant? Well, effectively, because we're going to 365 days green, and we're going to pre-monsoon sowing strategies, we are obviously already made the decision, no, we are trying to plant before the right season, and then it's a question of how protective that pellet is for the time till the season is right. See, so we don't have to sort of really select, this will germinate in November or this will germinate in August, but we do have to say it's got enough protection that if it is only going to be in August and we're sowing in May, we've got 4 months, 5 months worth of protection. There's no point putting pellets out which we know will only last 2 weeks when it may be 3 months before the next rain.